Welcome to our virtual comparative genomics workshop. The Institute of Food and Agricultural Sciences, the University of Florida, and the National Science Foundation sponsor our research group to give 2 or 3 days workshops covering this material at the University of Florida and other institutions. IFAS, UF, and NSF have also sponsored the creation of this series of video lectures. This is the third in our series of videos, and it is entitled Using Phylogenetic Distribution Tools. The material covered in this lecture was prepared by Drs. Andrew Hanson, Valerie de Crecy-Lagarde, and Claudia Lerma Ortiz. In this lecture, we will describe briefly 3 platforms that offer phyletic analysis and identify their operational facilities. We will then proceed to explain the principle of phylogenetic distribution and give you instructions concerning the use of these tools for the search and identification of candidate genes coding for an orphan enzymatic activity in a well-defined group of organisms. In order to illustrate the application of these tools, we will give you some examples taken from the research on folate metabolism. We will also cover, albeit very briefly, methods to refine our search, like the use of the string database. For more information about string, please watch our second video. Finally, we will round up our lecture with a comparison of 3 different platforms. To train you in the operation of phylogenetic distribution tools, we will use the following. The Phylogenetic Profiler operated by the Joint Genome Institute. The Phyletic Pattern created by the Microbes Online team. The Phyloprofile Exploration offered by the Microscope Group. Both the Phylogenetic Profiler of the Joint Genome Institute and the Phyletic Pattern of Microbes Online are supported by the Department of Energy of the United States of America. The JGI Phylogenetic Profiler is operated by the University of California for the DOE. The JGI Profiler website has the address shown on your screen. The Phyletic Pattern of Microbes Online is a creation of the Virtual Institute for Microbial Stress and Survival as part of the U.S. Department of Energy Genomics, GTL program. The Phyletic Pattern of Microbes Online has the web address that is shown now on your screen. The operational principles of this platform are described in the paper entitled Microbes Online, an integrated portal for comparative and functional genomics, by Paramveer S. Dihal et al. And published in Nucleic Acids Research in Volume 38 of the 2010 publication. Database issue, pages 396 to 400. The Phyloprofile Exploration was created by the France Genomique Group. This group is formed by Le Commissariat à l'énergie atomique et aux énergies alternatives, CEA, l'Institut national de la recherche agronomique, INRA, le Centre national de la recherche scientifique, CNRS, l'Institut national de la santé et de la recherche médicale, INSERM, le Centre européen de recherche en biologie et en médecine, IAI, and l'Institut Pasteur, and la Fondation Paris Sciences et Lettres. Please note their website addresses shown on the screen. We will now explain the phylogenetic distribution tools, operational principles, and give you step-by-step instructions on how to use the three platforms.
We're going to teach you a tool, one of the phylogenetic profiler tools. So we're going to learn this with an example, and I'll explain the principle first, and then we'll go to the example. The principle actually goes back to this principle of the phylogenetic tree that we drew, schematically in the PowerPoint presentation, where, some organisms have a gene and some organisms don't have a gene, either plus or minus. The idea is that if this is a biochemical pathway and that these three branches of the phylogenetic tree have the pathway and these two don't, or these three don't, then all the enzymes associated with that pathway should be here and all of them should be absent there. It's an oversimplification, but that's the principle. Suppose that we already knew several of the enzymes in this pathway, but we didn't know all of them. Well, we could pick any one of the enzymes we did know and predict that all organisms that have the pathway will have that enzymes, and then those that don't have it won't. So we can find an enzyme in the pathway that's missing. We can look for genes that correlate exactly with the distribution among organisms of the known enzyme. So in other words, you're saying the unknown enzyme, if there is one, a missing enzyme in the pathway we haven't discovered yet, will simply track in its distribution that of the known ones. Anybody got any difficulty with that basic principle? Really, that's an easy way to think about it. The reciprocal is also true.
And the reciprocal, teaching that for a long time, you guys have a lot of trouble to get it. So it looks easy, but think about what he says now.
Well, he has a lot of trouble getting it. Remember, I'd have to call you and ask how to do it. Yes, this is challenging. The reciprocal is also true. It's probably easiest to think of it in terms of, say there are two different versions of a pathway here. This, we know the pathway is present in all the organisms. We know that it is present in these. We know that there's an alternative version there, but we don't know the genes for it. So we know they all got the pathway, but we know that these, and we know the gene. In this case, we don't know the gene in the open case. Now in that case, in order to find the gene that, if you like, replaces this one, we look for something that has exactly the opposite distribution to those. Instead of it being present in the genomes, we ask for something what's absent in these genomes which have this gene and present somewhere else. So you're asking for the flip side of a distribution.
The entire success of the research done using phylogenetic distribution tools depends on the careful selection of the bacteria that will constitute each of the two groups needed for the analysis. In the particular case that we just mentioned, we can apply the information from phylogenetic trees to choose representative bacteria from different clades. Then we may use different platforms like NCBI and SEED to do blast searches and confirm the presence or absence of the B gene homologs in all the representative bacteria that we choose. This practice will ensure correct allotment of the bacteria to the groups having or lacking the B gene. We should also take advantage of these tools to make sure that the bacteria in both groups have the metabolic pathway in question. We can do this by checking the presence of the other genes of the pathway that are commonly present in both groups of bacteria, like genes A, C, and D in the current example. Usually, about 5 to 10 different bacteria are selected for each group. Once this careful selection has been made, we enter the names of all the bacteria into the phylogenetic distribution platform of our choice in the required form. Oversimplified, the phylogenetic profiler analysis can be explained in the following way. After we load the names of the bacteria, the information of these bacterial genomes is retrieved from the platform data bank and two different gene lists are generated. First, we will concentrate on the list of all the genes present in the bacteria which hypothetically have the incognita gene represented by Q. For the sake of simplicity, we are only using two bacteria, U and W, to illustrate our example. We know that U and W bacteria have known A, C, and D genes, so we may identify the genes in this list. We have hypothesized the presence of Q, but since it is unknown, we have no idea of its location in the genome and it remains hidden in the list among thousands of other genes. Let us now concentrate in the second list containing all the genes of the bacteria that have the B gene. In this case, we know that these organisms do not need the incognita Q present in their genome in order to have an active functional pathway, and hence, in general, we are hypothesizing that they do not have the Q gene. Please note that this is only an hypothesis and that there might be a few bacteria that do have both genes. If we inadvertently choose for our research a majority of bacteria having both alternative genes Q and B in their genomes, we will get equivocal results which will force us to select other bacteria to form new lists and start our research afresh. Please look carefully at our list. X, Y, and Z bacteria are missing genes 3 and sigma. Moreover, among the other thousands of genes in their genomes, they might be missing some other genes that could be present in the U and W bacteria. Keep that in mind since we will revisit this case and explain how to solve this problem later. Please also note that these bacteria have genes A, C, and D, and hence they should have a complete metabolic pathway. Well, so the next step is for the phylogenetic distribution program to process the information contained in these two extensive lists and digitally compare and contrast them to finally generate a list of all the genes present in both groups of bacteria ordered by homology. This list could look something like this, and it is composed of thousands of genes. However, the program is able to extract the necessary data from this list, producing a final table containing all the genes that are present in the group containing the incognita Q, and absent in the other group of bacteria containing the B gene. Sometimes we could get a final list comprising more than 50 genes, most of which could be coding for unknown proteins. In this case, we might have to go back to the drawing board and make a second selection of bacteria to construct new groups of analysis. Other times we might get around 20 genes, most of which could have already been well characterized with only 4 or 5 exceptions. In this case, we would be able to apply other tools to find which of the unknowns is our most feasible candidate to be Q. For example, we can use the PubSeed platform to analyze clusterings of the candidates, or the string database to look for co-occurrence, fusions, co-expression, and other information concerning these unknown genes relationships. Fortunately, an appropriate combination of these tools can enable us to narrow the search to 2 or 3 final candidates. In the following examples, we will use a case where we are able to get a single final candidate Q, and other cases where we get more than 5 genes from which we then have to choose the most feasible candidate.
It will come clearer when I give you a specific example, because it's pretty self-evident how you'd look for positively correlated genes among organisms. I mean, that's self-evident. It's looking for the flip correlation that's a bit more challenging, and if you can do the flip correlation, then you can automatically do the positive correlation. So we're going to teach you the flip one. with a specific example which corresponds just to the thing I've drawn there, schematically. If you look at this column, look at this basically phylogenetic tree of life. This enzyme here, YGFA, this enzyme is required to dispose of a toxic product of folate metabolism, let's call it that. That product is formed by all organisms, so all organisms have the problem of getting rid of this toxic folate product. Therefore, it's a pathway that's everywhere. We know it's actually only a 1 or 2 enzyme pathway, but we know the pathway to get rid of this product must be everywhere. We know the commonest gene that carries out this pathway, it's this thing YGFA. So YGFA is nearly everywhere, but not quite everywhere. There's some genomes rather spotty in terms of its taxonomic distribution of these genomes. There are genomes that don't have YGFA, but we know they have the pathway. So they must have something else. Well, it turns out that what they have something else is a gene that we're calling here, we're calling it COG3643. So if you look down the distribution of it, this was something that actually I found this manually with help from a subsystem that Ross Overbeek, the genius who founded the seed, had built years ago. So it's really us together in a way, although it was separated in time. I found that manually that this gene was pretty well always present when the known gene was absent, and that regardless of taxonomy. There were also some cases where they were both present. But as I was telling you a little while ago, that shouldn't worry you. Remember E. coli with the 2 genes that do the same thing, the animal type and the plant type? A few exceptions don't worry you. So, although I found this by dint of manual analysis using Ross Overbeek's subsystem, it really is obvious from this that you ought to be able to do it computationally by looking for a gene which is present when YGFA is absent. And as long as you don't make it too rigid of a correlation you're looking for, as long as you allow some slop in it for cases where they'll both be there, you ought to be able to compute just by scanning through genomes. If you don't have that genome, you ought to have whatever replaces it, and that will be the distribution pattern of this gene. We predict it to be present when YGFA is absent. Yeah? If anybody doesn't get that, we'll go over it again.
Wouldn't it be possible for another gene that serves the same function to be acquired horizontally, for example, and then use the original, so you end up with a different gene than that one substitute serving the same function? Maybe not in this system, but in another system, say.
Well, this COG3643 that replaces YGFA is not any kind of a homologue of it.
Yeah.
It's a completely different biochemistry. It just functionally does the same thing.
I'm just saying, like, wouldn't it be possible to have another gene that's not a homologue, that serves the same function, but that's different from one of the substitutes, so that you might end up with different substitute genes that have no homology with one another?
Oh, you're saying that there could be more than one solution to the problem.
There is.
Amen. That can happen.
That happen. It doesn't. It not can happen. That happens. That's why designing the profile is difficult, because you don't know where that happens, and so you always try different profiles.
But the good news is that there are some kind of reactions that are fairly difficult for biology to do, so it doesn't have a lot of solutions. Now others, like I can tell you phosphatase type reactions where there are definitely members of different phosphatase families carry out that step in different organisms, and it would be very difficult to find them with this kind of global analysis because they're local solutions. The key gene is missing in this group, there they replace it with one kind of phosphatase. These people would do it with another kind. So yeah, you're on the ball completely. You've hit at the probably the one largest difficulty in using this approach, but it's, with all that said, it's surprisingly often fruitful. And the thing that I'm going to show you now with this example, it's not an artificial example. I mean, when I'd done this manual discovery, I just tried the tool to see if it could replicate it, and it did it in about half an hour. It took me that long just to do it. Well, I'd wasted about 2 months trying to do, well, not full time, but a couple of months of serious study when I could have pushed a button and had the answer in half an hour. So it's much better to look for the answer and see if anything pops out before you do a deep, manual analysis. And Valerie's had great success with various kinds
I'm the champion of this.
Because again, you're dealing with quite complex chemistry that life hasn't invented lots of different solutions.
But it's not, you have to try lots of different profiles. It's not like the first one you try works.
So there's more than one tool to do this. We're going to use the one at the Joint Genome Institute, the DOE JGI, and we're going to look for a gene that has the opposite distribution to YGFA, which is the characteristic that we predicted would be but the gene that we want. So look at the genes that we're going to use as our profile. In other words, the ones we're going to define as having YGFA or not having YGFA. Now, the first thing you have to do in all these profiling programs is you have to select one genome to report the gene that's missing.
Actually, not all of them, only IMG. The others, you don't need one. But
really?
Yeah, microbes online, you don't need one.
Okay.
It does,
I stand corrected.
Yeah.
In this one, you have to select one genome to report the missing gene. So that's going to be obviously a genome that doesn't have the known one. Okay, so we've basically taken 5 genomes that don't have the gene, one of which we've selected to report, and we're going to ask for genes that are present in that and don't have homologs in the ones that do have YGFA. So we've got 5 genomes here that do have YGFA, 5 that don't. We're picking one as the reporter and saying, look for genes that are in this organism that are not in this organism. Now this is the mental, difficult bit, because you're asking to find a homologue of something hypothetical.
Yeah.
That's the thing that I find the toughest. But you're working basically by asking for a gene which is not in the genomes which have the known gene. Okay, so we're now going to go to this spreadsheet.
So that's where it's difficult, because if you have a genome here that has both, you'll never find it. Think about it. If you have a genome here that has both this one and this one, and you do that query, you will not find it if your criteria are strict. You might find it if you loosen.
You find it pretty successfully if you use the default criteria.
Yes.
Actually. Because this isn't asking for absolute.
It's not 100%.
It's allowing you like 80% correlation.
Yeah, they can be present in 4 out of these 5,
Yeah.
or absent in 4 out of these 5, and you'll still get it in the list.
Yeah.
But if you change that and make it 100%, and you have one of these that has the 2, the one that we know about and the one that we don't know about, you won't find it.
I mean, you've got a long list of genomes to choose from in that. Remember my picture? Most genomes have got this gene, so you could pick pretty much anything. Oh, that's another point. You've got a big interest in picking diverse genomes here as well, because the more you do that, the more you're going to get away from things that are just missing because of taxonomy.
Yeah, you'll reduce your list by doing that.
So you should, if you can, you should pick genomes that are as taxonomically diverse as possible, that do and don't have the gene.
To find written instructions for the phylogenetic distribution exercises we do in this workshop, type 1C page 1 in the Google search window. The first option that appears in your screen is the lab webpage of the Horticultural Sciences Department. Click on this link. You are now in Andrew Hansen's group webpage. Click on the Lab tab. Now look for the Workshop 1 link inside the red bar and click on it. A menu offering you different options will be shown on your screen. Please click on the Days 1 and 2 link. Once you are in this web page, please scroll down until you find section 10 of the workshop. In this section, you will find written instructions on the phylogenetic distribution tools. Scroll down until you can see under the heading Using the Phylogenetic Profiler at JGI the two lists of bacteria that we will use for our first exercise. Now you will need to open in a new tab the link for the Phylogenetic Profiler, which is given just above these lists of bacteria. You will have to be circulating backwards and forwards between our workshop page and the Phylogenetic Profiler tool so you can feed the info into this platform to complete the two lists of bacteria needed for the search. Let's start now.
So now we go to the spreadsheet itself. Keep your class open because we're going to be using those genes. And you've got 3 columns here. You've got find genes in, with homologs in, without homologs in. And what we're doing here is we're finding genes in just one organism, our reporter, which was Thermatoga, with homologs in all the things that don't have YGFA and without homologs in all the things that do have YGFA. So the first thing we do is look for the organism where we're going to look for the genes in, which was in Thermatoga maritima. So look for, using the search tool in your browser, look for Thermatoga maritima. And there are actually 2 of them. It doesn't matter which you use. You can use either of those.
To find your genome, doing the Apple F is a trick, because otherwise you have scrolling down and finding your genome. So
yeah, so use the search tool in
Search
the browser
tool, yeah.
to find it.
Yeah.
There is no other search tool. One of these does have a search tool in it, doesn't it?
yeah, you can just type it.
Type it in, but
like
this
microbes
one
online.
this
That's
is-
why microbes online is the easiest to use.
This is clunkier, but it works. Okay, so find Thermatoga. I would actually use the first one, the one that came up there. Thermatoga maritima MSP8, and then put the radio button on in the extreme left-hand channel. And now, everybody listen please. That's the only one that's going to be lit up in that column. That's the one which is our reporter. Now we're going to put the 4 other ones which don't have the gene in this column. So go back to the days 1 and 2, and it's Streptococcus pyogenes M1 gas. So if you pick that up, put it in the search box.
I actually find that the best way to find them is to put the strain number, because that's a unique identifier. You have to put it in Control F.
Oh, thank you.
Command F, and then you can put it. And I would put just like the strain name, gas, or whatever, because you're going to find them easily.
Okay,
that way.
thank you. Yeah.
They've just sequenced 200 strains since we did the tutorial. That's the only problem.
Actually, they've just added a whole lot
Yeah.
of new M1
Yeah.
gases. I don't think it matters
Yes.
which one. I did this at the weekend and it worked with the one I picked. So remember, these you're now clicking off in the second column. So has everybody now put 4 genomes in the second column? Anybody not done it? Okay, so if everybody's got their 4 genomes in this column, the next column we're going to populate is the 3rd one, and we're going to populate that with genomes that do have YGFA.
Okay, this column.
So now we go back to our spreadsheet, and we've got the without homologs in here, starting with E. coli K12 and then down to Treponema pallidum SS14. But remember, these go in the 3rd column, not the 2nd column. when you do E. coli, pick MG1655. That's the standard strain. I don't think it matters, but you should pick that. And remember, third column, not second column. It says without homologs in.
Means they don't
that
have
helps, yeah.
YGFA.
Aha, you see now,
Huge.
you're with me. I mean, this is the problem I have.
That's
Because
what I told
you're
you.
asking for this mythical thing that you don't know exists. So you're asking it for, we're saying that if you've got YGFA, the one we know about, you won't have the unknown one that we're looking for. So that's why you're looking for something without homologs in the genomes that do have the known gene. The hypothesis is if they've got the known gene, they won't have the hypothetical unknown gene. So we're looking for something that has no homolog in E. coli because we know the gene that does it here.
If you've done some mathematic reasoning, it's the absurdity reasoning if you do the math. Raisonnement par l'absurde. It's exactly that. If you understand-
You're postulating an abstract entity that is not present where we already know how the pathway works. That's
But you're making a big assumption is that these things don't have the two pathways.
that assumption is more often true than not, which is why this will
Yeah.
work. So when you put all those in, go back to your phylogenetic profiler. At the bottom of the page there's a go button, but before you hit the go button, there's some things, we're not going to change them, but you can relax this or tighten it. You can increase the E value, which is what tells you that the gene is there. Well, -5 is a pretty weak homology, so if you wanted to be more discriminating and not pick up vague homologs, you might want to push that up. And then minimum percent identity, that's another criteria similar to E value. And then you have some algorithm choices. But anyway, hit the go button when you've done it.
So IMG has one thing that's really cool is it gives you the exact result of the search. And that's actually, when I do this, I copy this and paste it in my notes because then it's kind of a trace of what was done.
And at the end, it reports actually nowadays it reports 2 genes. When they were putting, first of all, for a couple of years, then there were fewer genomes, it was only reporting the one, which is the good one. Now it's reporting the one that's the good one with the higher score. And this is typical. You usually get a couple of candidates out of these things, but let's say that we didn't know.
No, he's dreaming. You get a couple of candidates when he gives you this very well-designed query for a class. Yeah, when you start on a real project, you'll get hundreds. Yeah, and then
No, I don't get hundreds. I get 5 or 10 maybe.
yeah, but then you can miss those. So you have to start. You go down, but you shouldn't. When this happened really only once or twice in real case.
But I didn't have to fake this in any way. I
Oh
mean,
well, come on! In your choice of your genome.
this is exactly what I did. And to my shock and horror, having spent 2 months finding it the hard way, it just came right out this way. But no, seriously, there will be a prioritized list. In general, the priorities are less. The priorities that it gives you are less significant than the annotation it will have, because sometimes that will tell you right away, this is interesting. And just put yourself in the shoes of the investigator who's looking for a gene that replaces a folate metabolism gene, and then you see this 4-aminotransferase cyclodeamidase, 4-amino tetrahydrofolate, and right away the annotation is telling you that, hey, yes, this is something to do with folate. So this turns out actually to be a gene which is known to have another function, and it doubles. It takes on a second role, a moonlighting role, they say, in folate, in removing this toxic product. So,
You're interested in the family. You want to know the family that replaces the initial family in these organisms.
In all of these organisms and more. We've just picked a representative half dozen.
And so that's a group you're working with, where you know the pathway and so on.
No, you know that these organisms don't have the regular enzyme, but they have the need of it, because he didn't discuss, but if you don't need this enzyme, you don't want to have it. So you have to have some biological information that these organisms will need that enzyme, or because you know that the rest of the pathway is there, or you know that they're not oxotrophic for this thing, or you know that in that case the problem that this enzyme fixes occurs in these organisms. So usually you have to add a layer of biology in your choice.
The enzyme in question that's replaced, these have this YGFA. The reaction that YGFA carries out, or the compound that it disposes of, is everywhere. All organisms make it, so you have to deal with it. That's a given in this case. The organisms that don't have it, when you look at this phylogenetic distribution, the organisms that don't have the red box, they have to have something. It's like, think of it, suppose you were missing
Is everyone on
pyruvate dehydrogenase, everything's got to have that.
Yeah.
And because they all, any aero.
Okay.
So it's got to be there, and it's got to be something.
Okay. So I'm going to show you how to use other phylogenetic profilers. We can do it on the same example, or we can do it on the Phyli1, Phyli2 example. So if you go back to the Hansen lab first page in your workshop 1 in the resourcement, you have this phylogenetic distribution tools. So we've just gone and used the phylogenetic profiler at JGI. We're now going to use this Microbes Online one. So you have to go back to the resourcement page and find this link. So at one point or another, you have to be on that page. So to the resourcement, on the resourcement page, the Microbes Online phyletic pattern, you copy that address, put it in a new tab, and you'll be here. So the difference with IMG is how this thing is set up, but the reasoning is the same. So here is the list of all genomes in this Microbes Online, and if you type in the first few letters, you'll have the genome that you're interested in come up, and you're going to pick it and select it to move it in your genome selection. So you're first going to put all the group that you want your genes to be in, put them here, and click Set Present. That will be a first group of organisms. You don't, in that system, you don't specifically put one on the side like the thermatoga. You put all of them in that group. Once you do that, you'll have a list of families that are present in these X organisms. It will be there. Then you do the same. You pick the ones in which you want the genes not to be present, and you move them to that, and you click Set Absent. And that will do the overlap between the two lists, and you'll have your list of genes. So you can try for another example, because you've already been exposed to the folate subsystem. In folate, you have the GTP saccharohydrolase 1 gene that you've heard about, FolE, but a lot of organisms don't have FolE, but they have the folate pathway. They have FolP, they have FolK, but they don't have the regular FolE gene. So here you have the regular FolE. You have all these organisms that have FolP and FolK and don't have the regular FolE. So here, these are the same query. So except in Microbes Online, you would just lump all these together. You wouldn't separate them out. So you can do what you want, or you try with this FolE2. I'll let you start and work on your own, because really it's the only way to get it. And then I'll do it up here to catch everything up.
Where does the set selected go? Is there a list somewhere?
Here, once you put here, you add and it goes here.
Right. And then you do...
You have the whole list, you do all of them you want, and then you click Set Present.
Right.
And then you do the same, Set Absent. You do it in two rounds. You can type the first letter, you can type the last. So the problem is, I don't know if they're going to have the exact same genomes. We're going to try. So the only thing that gets confusing is to hit the right button in terms of set present or set absent. You have to think. So I'm doing the Folly 2 example. For the strain name of Staph aureus in the Folly 2, use the call strain. I didn't put it in the document.
Here, we will guide you step by step in how to enter your data into the Microbes Online Phyletic Pattern platform. Type the abbreviation located at the end of each organism's name on the top left little window shown on the screen. For example, if you type MSB8, the name Thermotoga maritima MSB8 will be displayed on the big window below. Select this organism by clicking on its name. A blue bar enhances it now. Click on the Add button at the right of the little window. The program will automatically transfer the name of the organism into the list of the genomes selected displayed on the right side window on the screen. Repeat the same process to add the following bacteria to your genomes selected window: Neisseria meningitis FAM18, Bordetella bronchoseptica RB50, Geobacter sulforedescens PCA, and Staphylococcus aureus aureus. This is a group of bacteria which must have the incognita that is replacing the Fol E gene. So you have to click on the button Set Present. The program displays a list of 200 genes which are present in all these 5 bacteria. We are just showing here a few. Scroll down to see the complete list. We are going to compose the second list of organisms now. This list includes all the organisms that do not have the incognita gene, and they have a Fol E gene instead. Saccharomyces cerevisiae RM111A1 and Escherichia coli strain K12 substrain MG1655. Again, follow the same procedure as above to select the names on the list given on the left window of the screen and subsequently transfer them to the window on the right to compose your second list. Finally, click on the Set Absent button at the command's bottom right corner. This list comprises the set of organisms which do not have the incognita gene. The program finds a phyletic pattern from which it selects the genes which could substitute for Fol E.
So this is the one for Folly 1, Folly 2. You see the genomes are not very far. So this is a more realistic example where you're faced with reality, okay? So that's why it also shows you why I always use 2. I never use only one phylogenetic profiler because they give you the settings they have in their BLAST searches to define families are different. So here, the ones that did my Folly example and get to that, you did your survey right. However, now what you have here is a bit different than the output that you got from IMG. This is for every genome of your set present list: Maritimas, aureus, meningitis. And this is the list of families that are present in all these genomes and absent in the ones you set absent from here. These are, remember yesterday he talked about cogs or families. This Microbes Online gives names to these families. They're not totally homogeneous, but it's pretty much the same family. To know the exact specific gene, you have to click on that, and that will bring you to the member of that family in that organism. So the first thing you do when you're doing that in real, you see that all of these have annotations. And if you know your microbial biology, you actually can figure out what these do. This is histidylcyanide synthetase. This is the the Nudix enzyme. But then you have 2 which just have the cog number. So these are the ones you're going to go and look at first because they might mean that they're not annotated, they're just cogs. So you're going to go and look at them and see, do you have any other type of evidence that could suggest that it's your missing Fol E gene? So are they linked to folate when you do that. So what would you try and see? What would be your next step here? You could do a CDD search. That's one thing. I'm afraid that if I go onto it, it'll be annotated between the two. But you would do a CDD search, and there it would tell you t-fold unknown function. So actually, it was not really helpful to start. But what was helpful was the other type of things you can do. So this is the correct one. But what I want you to show you is that if you go to the gene, and then that's the trouble with microbes online, is all their stuff is very hidden. And if you go to the browse genomes by trees, you'll be brought to a nice feature of microbes online, which is their clustering tool. Remember, in seed, you have their clustering tool, and any annotation page, any Facebook page, you have your genome, compare genome view. This, if it brings us there, is pretty much the same view in microbes online. It'll give you the compare view here, okay? So here is this COG 1469, and what you see is the pink one is a full P gene. So you're clustered with a full P gene in a few genomes. So that's actually how I discovered this gene. I did a phylogenetic pattern. I had a list of 20 COGs, and then I looked at clustering, and only this COG clustered with folate genes. It was typically the reasoning we used. We had two types of evidence. One was a phylogenetic distribution query evidence. That's how we went to the first page. From there, I went through all the lists, and I looked at clustering. And I found that one of them clustered with folate gene. This is one example, but there are other folate genes in which it clusters. So this is a good example because a lot of what we do is combining different type of association evidence. Often one type of association evidence, you're like, but when you have two or three, we pretty much run to the lab. I got this clone from Thermotoga from a structural genomic effort. They send me the clone, and we put them in a Delta Foley strain, and we had our data. Our complementation. So when you have several lines of evidence, your confidence score in your prediction really goes up. You know, it goes from maybe to I'm nearly sure. So I want to make sure that you are all able to do this. So this is why I always use two different profilers. They give you different results. Because the way they define their superfamilies is different, their cutoffs are different, and you don't really have any mastering of that. So when I look for profiles, I do 2 or 3 different profiling programs, and I compare the results. If one is found in the 3, it goes up in the list, but if one isn't, it doesn't kill it.
Also, sometimes it's worth looking like on IMG, looking at the output, you know, the individual
Yes.
lines of what genome was subtracted,
Yes.
you know, as you go.
Yeah.
Sometimes it helps. You can discard one
Yeah.
if you have too many genomes.
Yeah. So here it must be, there's 2 reasons, or we didn't use exactly the same strain or in their genome, or the way they define family is a bit more stringent and we're not getting it. This thing has paralogs, so if the paralog cutoff was higher there, you're going to take it away. But I want all of you to be able to get to this one, okay? I want you to do it on the second example. So the problem with phylogenetic profiling, it's very easy when we give you the right set of input genomes. The difficulty is figuring out the input genome.
The string database is another tool we could use to pick the candidate gene that could probably substitute the Fol E gene out from all the COGs that we found with the phylogenetic distribution tools. To show you this application, please go back to the Resource One web page and click on the string link. If you do not know how to use the string database, please watch our second lecture for instruction. Change the program to COG mode by clicking on the COG button. The display should turn pink. Dial COG 1469 and click on Go. Repeat operation for COG 1636. On the top is the summary for the results for COG 1636 and on the bottom for COG 1469 concerning neighborhood, fusions, co-occurrence, co-expression, and other types of relationships of the queries with their functional partner COGs. From the results obtained with string, we conclude that COG 1469 has relationships in both the ambits of gene fusion and neighborhood with folate metabolism genes. Hence, COG 1469 is the most likely candidate to substitute FOL-E. As a training exercise, you might want to use the Microsoft Online platform to search for the gene that substitutes YGFA. As we mentioned before, different platforms have different gene databanks. So in this case, you should modify the list of the organisms having the incognita gene Q using only Streptococcus pyogenes M1 gas, Streptococcus pyogenes SS1, and Elusomicrobium minutum P191. You can use the same list for the organisms which have YGFA and do not have the incognita gene Q. Escherichia coli, strain K12, substrain AMG1655. Streptococcus pneumoniae D39, Streptomyces coelicolor A3, Aquifax aeolicus BF5, and Treponema pallidum SS14. Following the method described above, you will arrive to the results shown on your screen. As you can see, this analysis renders a list of 7 putative genes that could substitute YGFA. Let's analyze this table to discard genes which are obviously unrelated to folate metabolism. Clicking on the links for each candidate gene, we realize that RGPFC is probably related to the chain formation of polysaccharides, and COG 1518 is related to DNA repair, so we may discard these from our list of candidates. On the other hand, COG MIS1 is suggested to synthesize formyl tetrahydrofolate. This is the reverse reaction of the protein we are trying to identify, so we will make a note and discuss this later. Finally, COG 3513, COG 1636, COG 3512, and COG 3643 are all unknown proteins, so our candidate Q might be any of these. Using string to analyze COG 3512 and COG 3513, it can be seen that they are related to each other and to bacterial phage interaction and have very similar string profiles. And COG 1636 is related to peroxyredoxin and other COGs, but not to folate metabolism, so we may cross all these COGs out of our candidates list. COG 3643 is another story because it has relationships in the ambits of gene fusion, neighborhood co-occurrence, and co-expression with folate metabolism genes, so this might very well be one of our chosen candidates. Remember, we had already identified precisely this COG as substituting YGFA by using the JGI profiler early in this lecture.
Try and figure out how to do it, to do the same query, do it in the third profiling site. Microscope. So it's at the Genoscope. This is one of the French sequencing center. And they have integrated database who does also phylogenetic query. And I think here there's a trick, if I remember well. Let's see. I think here you have to define an input organism, but it's tricky where you define it. So I think here, by default, it's set to Acinetobacter bailei, and you want to change that to Thermotoga. That would be the organism in which they're going to give you the result. So it's hidden up there. That's one of the tricks of that one. So you would change that to thermotoga, and then find genes with homolog in that list, and they have different lists. They have their own list, and they have the NCBI list, so you have to see which list has the genomes you want.
Here are our step-by-step instructions on how to use this third platform. Please go back to our Comparative Genomics Workshop webpage and click the link for Resource More. Scroll down to find the link for the phyloprofile exploration of microscope and click on it. You are now in the microscope main page. We are going to repeat the first search example using this new tool. Remember, in the first example, we looked for an incognita Q which had the same functional role as JGFA. You can see on your screen the two lists of bacteria with and without the incognita gene Q that we are going to use in this example. They are the same sets that we use with the JGI profiler and are also given in our instructions for the workshop webpage. Now, let's take a look at the microscope page. Please note the big black toolbar on the top of your screen. Notice the little button located on the top right corner of the bar with the word Change on it. Click on it. A new window is displayed on top of the microscope main webpage. This is the Reporter Choice page, and it shows a list of bacteria with a little search window underneath. Notice Actinobacter bailey has been automatically selected as reporter gene. We are going to change this by doing the following. Type Thermotoga maritima in the small search window. As the complete name appears on the list given above, click on it to select it. After it is highlighted with a blue bar, click on the Set Selection button at the bottom of this window. Automatically, the microscope program changes the name to T. maritima in both places, at the top right corner and at the center of the page under the title Phyloprofile Exploration. Please concentrate now on the pink side of the microscope page. We are going to construct the list of bacteria that lack the incognita gene using the NCBI RefSeq list by following these steps. First, type the letters at the end of the bacteria name BF5 on the spyglass window. The name Aquifex aeolicus BF5 is automatically shown on the top window. Click on it to select it. The name is highlighted with a blue rectangle. Now proceed to type MG1655 on the spyglass window to get the Escherichia coli name on the list above. Select it and proceed the same way with the rest of the bacteria to fill up the lists of organisms lacking the incognita gene Q. The list should appear complete on the Pink Zone's NCBI RefSeq list. The next step is to construct the list of bacteria which hypothetically have the incognita gene Q using the NCBI CEF-REC list of organisms at the bottom of the pale blue zone displayed on the screen. Start by writing 191 on the spyglass window to get a Lucimicrobium minutum P 191 on your list. Select it and so on, following exactly the same procedure that you used to complete the first list until you complete this second list of bacteria. Remember not to put the T. maritima on this list since you already assigned it to be the reporter gene. Once you have completed both lists, check them and scroll to the bottom of the page. Change the settings to homologies in all selected sequences by clicking on the corresponding radio button and then click on the View Comparison button at the end of the page. the phylogenetic profiling analysis will be displayed at the bottom of the page. The first alternative on this results table is the formiiminotransferase cyclodeaminase that may also act as formiimino tetrahydrofolate cyclodeaminase. This is the most plausible candidate to substitute YGFA, and it is the same gene identified by using the JGI phylogenetic profiler earlier on this lecture.
Try and figure out how to do it, to do the same query, do it in the third profiling site. Microscope, and try and see if you can find the missing Phyli gene. Do the Phyli one, the second exercise, example 2. So the third one you can try is this microscope. So it's at the Genoscope. This is one of the French sequencing center. And they have integrated database who does also phylogenetic query.
Let us now use the microscope program to identify the substitute of Phyl-E. For this exercise, we will also use T. maritima as our reporter gene, and we will use the organisms whose names are displayed on the screen at the moment to fill up the selections of with homologs in and without homologs in the NCBI SecRef lists respectively. To wit, in with homologs in Bordetella bronchoseptica RB50, Geobacter sulfur-reducens PCA, Neisseria meningitis FAM18, and Staphylococcus aureus RF122. In without homologs in Arthrobacter SP FB24, Bifidobacterium dentium BD1, Bifidobacterium longum DJO10A, Escherichia coli K12 MG1655, and Cucuria rhizophila DC2201. If you follow the method we just showed you, you will get a final table with 5 candidate genes. Notice that the very first of the list is the GTP cyclohydrolase Phyl-E2. This gene is now called Phyl-E2 after the research and publication done by Dr. de Crécy-Lagarde and collaborators, but if you had done this search before, it would have just been called a hypothetical protein.
Frankly, microbes online is easiest to use. So my experience is JGI is the most updated in genomes, and it has the eukaryotic genome, which is very important because it's often when you do negative, you put the eukaryotic in there. The one I actually like the most is Microbes Online. This one has really nice metabolic tools behind it. The microscope. So the two things that are very clunky with this one is changing the reference organism up there on the left, on the upper right, and the fact that you have to use your shift and go down and find your organism, you can't search. What? Command. Your command, and you select the ones you want, and then you add them as a group. So remember here, you're finding a gene that you don't know anything about in terms of sequence. It's not by similarity of things you know. So it's a very powerful non-homology based criteria. Homology is used to define the families that follow a criteria, but you're not asking anything yourself. You're not saying it has to look like GTP cyclase 1 or it has to look like something else. You're just asking for a distribution. It's a logical operation that you're doing.
But at the moment of initiating the search, it's an abstract entity that you're looking for.
Yes.